is what you're referring to right now morphogenesis or morphogenetics?
Exactly.
And what would be the difference between those two?
Well, they are the same. Morphogenesis is the process, morphogenetic process is the process whereby you create form, you create shape. And I think we know very little about this process. But we know that it's not in the genes only, that the genes are being used to create forms.
So, speaking about morphogenesis, on page 438, I believe, but from your book called The Principles of Development.
Oh, yes. Oh,
yes.
Wow.
You talk about the imaginal disc, which comes from ectoderms of insects. So what are imaginal discs?
So imaginal discs is in a way, it's very fascinating in the history of developmental biology or biology. It's a remarkable thing. Insects are very weird, the way they develop. Imaginal disc, everybody has seen the chrysalid, the pupa of a butterfly or of a... So normally out of the egg of an insect comes a little creepy crawly, a little slug that feeds for a few days, okay? And hidden in that little worm-like structure, there are very small bags of cells, which are called imaginal discs because of the meaning. We are not going to get into the meaning of the word. And those little cells that are growing as this little creepy crawly is feeding itself, and they are growing, okay? And there are about 10 of these discs, and every one of them is the seed of a part of the adult butterfly or the adult life. When this creepy crawly goes into forming the chrysalid, many people must have had silkworms, for example. People that like biology, they grow. So they can see that all of a sudden the worm goes into this chrysalid, which is what is used to make silk. And then there a fantastic transformation occurs in which the cells of the creepy crawly die, and these things become like a Lego. They become assembled into the butterfly or into the fly. The imaginal disc, so there is a little group of cells that will give rise to one wing, another group of cells that will give rise to the other wing. There are 6 legs, and every leg comes from a group of cells that sort of grows and has instructions to make a leg. And in that pupa, in that chrysalid that this assembly is happening. And then it ecloses and you get this wonderful animal. And that's the imaginal discs. It's a very it's the basis of a lot of the insects that we know. Particularly these insects like butterflies and flies that we have around, and they are remarkable because you wouldn't think that an organism is made like a Lego. And you might ask, how do the cells know how to assemble together? Because in the creepy crawly they are completely separated from each other. And all of a sudden, in that pupa that you must have seen in many places, and that some of our audience will have seen when they make the silkworms, everything magically, and now we can film these processes and we can see how the cells, because part of every leg comes from a totally different imaginal disc, which is these clusters of cells, and they all come together in a fantastic manner during the formation of the adult organism that is that fly. When you look at the development of animals, what you find, it's a big variety of modes of operation, of development. One of the things that we have learned over the last 20 years is a fantastic story, which I think we don't yet understand, which is that the genes that make us are not very different from the genes, that make us, sorry, I don't want to recant myself. The genes that we have are not very different from the genes of the fly. It's how the cells use those genes that create a difference between a fly and a human. It's not the genes. We are not very different in terms of a gene repertoire.
So the genes will give the raw material to the cell, and the cell then has to do something with it?
That's my view. That's the way. That's what I'm suggesting. A change of perspective, that we need to look not at the genes as the masters, but as the servants, if you wish, to the cells.
Would it be that the genes are like what's providing you the corn and the wood and the steel, and then you as the crafter are the cell?
They are the corn. They are the corn. Exactly. They are the corn. They are the steel. That's what they are. They code for those things. That's very clever, the way evolution has created this. And the cell, there is a very interesting relationship between the two. If the cell wants to make another cell, it's going to go into the genome and pick up as you said, the corn or the silk or the rope that it needs, and it's going to then make a...
Yeah, silk and the rope would be better analogies, because corn itself is a living organism.
Well, it's food, but the corn is food, and the cell also needs food, so it has to create food.
Right.
So I think that the cell, there is a very interesting symbiotic relationship between the two, but the cell controls the genome in ways that we are starting to see. Cells are able to sense how many cells they have around. They are able to sense pressure. They create shape. They use genes to do that, but the genes are not, as I said, there is nothing in the genes that say that we have to have 2 eyes or 5 fingers.
Again, for people who are listening or watching, you have a book that's for the general audience called The Master Builder, correct?
Correct. Thank you.
Okay.
Yes.
And then there's another book that you have with some other people like Walpart, which is published by Oxford University Press, called Principles of Development.
This idea this notion that there are features of the makeup of an organism that are not in the genes is something that I get into arguments because, as I've said, the biology is very dominated by a gene-centric view, all right? And I think what I find interesting, I've mentioned earlier that there is a new cadre of people that are basic physicists coming into the field, and they are much more prepared to think about these emergent properties and look for the cause of these emergent properties in a manner that I would agree with. But if you talk to the card carrier biologists, they would tell you that there is nothing that is not in the genes, that everything is in the genes. This is something that if I was to get into a room with 10 biologists, 9 of them would be very much against me, and we would find a very interesting discussion that I don't know what it would ensue, but I've been thinking something else that people sometimes get confused with what I say because they don't understand is this notion of epigenetics, which these days is very popular. Epigenetics, it's a concept that has evolved a little bit. It was first mentioned by Waddington, that I mentioned earlier, who was interested in theoretical biology and was interested in doing that. But the term, the way he expressed epigenetics was exactly in the term of needing to understand beyond genes and needing to understand the sort of emergent properties that are the consequence of the gene's activity. Today, epigenetics has become a proxy for modifications of the DNA that control transcription, which is a totally different concept. I call it the genetics of genetics.
Yeah, so you classify epigenetics as still under genetics when we're talking about that concept of genetic determinacy that we referenced earlier.
Right, yes. Yeah, and these days, actually, many people want to transfer all the things that genetics cannot explain to epigenetics, which, as I say, is just kicking the can down the road. I mean, this is what it is. But I do feel that epigenetics, in the original sense of Waddington, it's a very interesting concept that refers to the sort of emergent properties of the systems. Either they are systems of cells, or they are systems of tissues, or they are systems of organisms. As I say, if you got me into a room with a lot of my colleagues, they not the very close ones, who I think I believe and are willing to understand these things, but in most people will argue that there is nothing that is not in the genes, that everything in the end maps to the genes. Whereas I think that there is a whole world out there. This is something that I probably would share with Mike Levine, that we need to explore. I just maybe being a bit reductionistic and being a bit materialistic, I want to understand the basis of that. I think there is, at the end of my book, I discuss a very famous paper by Mark Kirshner and some of his colleagues from Harvard, which they call they try to advocate the need to explore in deep detail these emergent properties. They call it material vitalism. Vitalism is the idea that there was some missing force. But I think today we have a lot of evidence that these emergent properties exist in biological systems, and we need to understand how they work and how to harness them for medicine.
So there is something to vitalism in that there's something called material vitalism.
I mean, I discussed this paper. I refer to it at the end of the book. It's a very very interesting paper, because vitalism sometimes is used to an ad hoc explanation for things that we don't understand. But today, actually, we can see how things that we didn't understand emerge from the activities of cells or from the activities of molecules within cells. Okay, and this is something. I think that this is a very exciting century, because I think the cells, understanding the cell and what it does and how it interacts with other cells, is going to revolutionize not only our understanding of biological systems, but it's going to provide points of view to do with health and to do with a lot of the regenerative medicine. It's not going to come from genes. I think even cancer, now people are starting to realize that cancer, which in many places is the genetic disease par excellence, it's clear that it's not just the genes that can create cancer. In many situations, the genes are responding to the activity of cells. People would like to understand the cell of origin, as they call it. It's not just simply that you get a mutation in a gene or two genes or three genes and you get cancer. There is something else that changes in the cell that we still don't understand that leads to the changes in gene function.
I can understand how a cell can respond to a gene, but how does a gene respond to a cell?
So, as I said, very, very simple. I'll give you a good example from our work, and I hope I can make it clear. If not, I'll try another one. So we can culture these embryonic stem cells in a dish, in flat, okay? And we can tell them to do something, to create all the elements of an early embryo from anterior to posterior, okay? We are very organized. The embryo is very organized in the way it does it. So if we can tell them to do that, to activate genetic programs that do that, these genetic programs exist in a flat situation, they will do that. But they will do that in a highly disorganized manner, okay? Everything will be chaotic. All the programs will be there. We can measure them. We can see them. We can look at the temporal sequences of expression. Now, and this is part of our work, and what really bowled me over. You can make a ball of cells now that is about 1,000 cells of these embryonic stem cells and trigger the same program, okay? And if there are 1,000 cells, they will activate this program again very chaotically, very, very chaotically. Now we take 300 cells, 200, 300 cells, and it has to be very precise. We activate now the programs in those 300 cells, and they make an embryo, perfectly proportioned, everything. They are the same cells, they are the same genes. So what information have they got? And we know that the number is very precise. If we go by 100 cells or even by 50 cells, the thing doesn't work. So the cells are able of measuring somehow. We don't know if it's space, if it's numbers, what it is. We don't know what it is. But they are now starting to use the genome in a much more organized manner than if they are very many of them or if they are disorganized. This observation, which is one we made 10 years ago and is the basis for our research over the last 10 years, I find it fascinating. And I think we have systems to understand what is that the cells are reading. They are reaching now into the genome, but in a very organized manner. Right? Because their numbers seem to be able to influence. How do they do that? This is totally, I'm really bowled over. And the programs now become perfectly synchronized. They were very, very well organized.
I don't know what you mean by programs.
Oh, so a genetic program is that when your development starts, when development starts, the cell doesn't know anything, and it activates a gene cascade, This is what happens during development. You get a genetic program is like a program. The analogy is with computers. Many people would jump to me if I make the analogy, but in a way it's not that different. So you get one cell, it becomes two cells. And now in those two cells, you activate now a genetic program in one cell. So a gene is activated in one cell and a gene is activated on the other. That gene now will activate a set of genes downstream from it, because the gene in one cell is different from the gene in the other. Now the genes that are activated, and then you unroll a program depending on those genes. That's what I call a genetic program. In fact, in the book I discuss, but I think it's a vision that I have to help me, genes create time in development. This is also very interesting,
Genes create time.
Yeah, these gene networks, these gene programs create time because there are sequences of gene expression, right? And we know that they are very well timed. When we develop the great precision, these gene regulatory networks, as they are called, can create time. Cells create space. You see, genes cannot create, and I think it is this dialogue between the two, but in which the cells have a very big say, that we are seeing in these structures that we generate in the lab.